Learning goals. Welcome to this learning session on cell growth, mitosis, and differentiation. In this course, we shall address cell growth and mitosis basics, uncontrolled cell division, cancer, animal growth and specialized cells, plant growth zones and differentiation, key specialized plant cells. There will be a quiz at the end to test your knowledge.
Introduction to cell growth and differentiation. Cell growth, repair, and asexual reproduction are all facilitated by mitosis, a fundamental process of cell division. Following growth, cells can undergo differentiation, transforming into specialized cells with unique structures and functions. The mechanisms of growth and differentiation vary significantly between plants and animals, reflecting their distinct developmental pathways and life cycles. Understanding these differences is crucial for comprehending the diversity of life. The role of mitosis in organisms. Mitosis is the process by which a single parent cell divides into two identical daughter cells, each containing the same number and type of chromosomes as the parent cell. This process is essential for several key biological functions. Normally, cells divide by mitosis when new diploid cells are required for specific purposes. These include the overall growth of an organism, the repair and replacement of damaged or worn-out cells, and asexual reproduction. Mitosis in asexual reproduction. Asexual reproduction relies entirely on mitosis to produce offspring that are genetically identical to the single parent. This method allows for rapid population growth in stable environments, as seen in many insects and plants. For example, aphids, common pests of crop plants, reproduce rapidly during summer months through asexual reproduction. The offspring of a single aphid will be genetically identical and diploid, just like the parent, ensuring a quick increase in numbers. Similarly, plants like strawberries use runners, and onions use bulbs, both forms of asexual reproduction via mitosis. Uncontrolled cell division, cancer. Cancer is fundamentally a disease of uncontrolled cell division, where abnormal cells divide uncontrollably by mitosis to form tumors. Unlike normal cells, which adhere to strict regulatory mechanisms, cancer cells bypass these controls. Normally, cells cease dividing once growth is complete, or when repair is no longer needed. However, sometimes the intricate controls that signal cells when to stop dividing malfunction, leading to the uncontrolled proliferation characteristic of cancer. This malfunction often involves mutations in genes that regulate cell growth and division, such as proto-oncogenes and tumor suppressor genes. In animals, the journey from a single fertilized egg or zygote to a complex multicellular organism is a remarkable feat of cell biology. This process begins with the zygote undergoing repeated rounds of mitosis, producing genetically identical daughter cells.
Cells then continue to grow and divide, eventually differentiating into a vast array of specialized cell types. This differentiation is crucial, as it allows cells to adapt to specific structures and functions, enabling them to carry out particular roles within the organism, such as forming organs and tissues. Early embryonic development involves the formation of embryonic stem cells, which can differentiate into almost any cell type.
Examples of specialized animal cells. Animal bodies are composed of numerous specialized cells, each meticulously adapted to perform a particular function. This specialization is a hallmark of multicellular life, allowing for complex physiological processes.
Key examples include red blood cells, which are biconcave discs lacking a nucleus, specialized for oxygen transport. Egg and sperm cells, which are gametes designed for reproduction. Nerve cells, neurons, with their elongated structures, transmit electrical signals throughout the body. Bone cells, osteocytes, embedded in a mineral matrix, provide structural support. Smooth muscle cells, responsible for involuntary movements in internal organs.
Plant growth zones, meristems. Plant growth is localized to specific regions known as meristems, where cells actively divide by mitosis. These crucial zones are primarily found just behind the tips of shoots and roots, driving the plant's primary growth. After cell division in the meristems, the newly formed cells enter a phase of rapid growth and enlargement. This process is fundamental to increasing the plant's length and girth, allowing it to develop its complex structure. Apical meristems, located at the very tips of shoots and roots, are responsible for primary growth, increasing the plant's height and depth. Plant cell elongation and differentiation. Following division, young plant cells possess small vacuoles that play a critical role in their growth. These vacuoles absorb water through osmosis, causing them to swell and exert turgor pressure against the cell wall, leading to cell enlargement and elongation. This elongation phase is crucial for increasing the plant's overall size. After elongation, these cells can then differentiate into various specialized cell types, each with a distinct function. This continuous process allows most plant cells to maintain their capacity for growth and differentiation throughout the plant's entire lifespan. Specialized plant cells, xylem and phloem plants, like animals, rely on specialized cells to perform vital functions, particularly in transport. Xylem and phloem are two primary vascular tissues, forming the plant's circulatory system. Xylem cells form hollow tubes that transport water and dissolved minerals from the roots to the rest of the plant, providing structural support. Phloem cells, arranged into sieve tubes and companion cells, transport sugars, produced during photosynthesis, from the leaves to other parts of the plant, where they are needed for energy or storage. Specialized plant cells, mesophyll and stoma. Beyond transport, plants also have specialized cells for photosynthesis and gas exchange. These cells are crucial for the plant's energy production and interaction with its environment. Mesophyll cells, found primarily in the leaves, are packed with chloroplasts and are the primary sites of photosynthesis, converting light energy into chemical energy. Stoma cells, or guard cells, form stomata on the leaf surface, regulating gas exchange, carbon dioxide intake, oxygen release, and water vapor loss. These cells can change shape to open and close the stomatal pore, controlling transpiration. Specialized plant cells, root hair cells. Root hair cells are highly specialized epidermal cells found on the surface of plant roots, playing a crucial role in the absorption of water and mineral ions from the soil. Their unique structure is a key adaptation for this function. Each root hair cell possesses a long, thin extension, or hair, which significantly increases the surface area available for absorption. This extensive surface area allows the plant to efficiently take up water through osmosis and mineral ions via active transport, which are essential for its growth and metabolic processes. Lifelong growth and differentiation in plants. A remarkable characteristic of plants is their ability to continue growing and differentiating new cells throughout their entire lifespan. This contrasts sharply with animals, where most growth and differentiation occur during embryonic development and early life, with limited capacity for new cell types in adulthood. This continuous growth in plants is due to the persistent activity of meristematic tissues, which retain their embryonic capacity for cell division and differentiation. This allows plants to adapt to changing environmental conditions, repair damage, and continually produce new organs like leaves, stems, and roots, a process known as indeterminate growth.
Summary. Well done. You have completed the course, Cell Growth, Mitosis, and Differentiation. You should now be familiar with cell growth and mitosis basics. uncontrolled cell division, cancer, animal growth and specialized cells, plant growth zones and differentiation, key specialized plant cells. Should you need to revisit any of the topics in this course, please use the menu at the bottom of the screen.